Hello guys, today we are going to discuss about Hybridoma technology. Hybridoma technology is majorly used to create antibodies against a single specific antigen. Let us discuss this with some basics. We create monoclonal antibodies. Now, what are monoclonal antibodies? When you see the antibodies which are produced by a single specific unique parent B cell are called monoclonal antibodies. Now, monoclonal antibodies will attach only to a single antigen. Now, whenever you see an antigen, you will find a specific structures on the surface of antigen. These are epitopes, which will attach to the Y-shaped immunoglobulin called antibody on its paratope. Now, when you observe, whenever the paratope attaches to the epitope, you will see the formation of antigen-antibody complexes, which will cause the agglutination in the mixture, which will define the reaction specificity when you observe. Now, not all the antigens will attach to the same paratope. There is a difference and there is specificity. Now, let us see the steps in the Hybridoma technology. The first step is immunization. In this step, basically you're taking an antigen and you're going to inject in an animal such as mouse. When you inject, the spleen of the mouse will produce the activated B cells against that antigen, which will provide the antibodies against that antigen. But you're going to do this so many times, you're going to immunize so many times to ensure the high dose of the B cells inside the spleen. Now, after ensuring, the second step is to isolate the spleen from the animal. Now, you're going to kill the animal and you're going to isolate the spleen from the mice. Now, basically the spleen is present below the stomach. Now, it has actually already produced the antibodies against the antigen. You're going to aseptically remove the spleen from it. Now, after removing the spleen, you're going to enzymatically degrade it to isolate the activated B cells from it, which has already produced the antibodies against the antigen we have injected. Again, by using density gradient centrifugation, this will process. So this is the overall two-step process. The next step is selection of myeloma cell. Now, what are myeloma cells? See, we should fuse the B cells which we have isolated with the myeloma cells. Now, the next step is the selection of the myeloma cells. A myeloma cell, when you observe, it is basically a cancerous cell which has the high division property. Now, you all know for the cell division, you all need the nucleotide synthesis. In our body, it will happen in two different ways. One is de novo way and second is salvage pathway. Now, we are taking myeloma cells by mutating the HGPRT gene, which will block the salvage pathway. We are also mutating the immunoglobulin gene, which will avoid the immunoglobulin production, which we don't want. Orally, by this step, you will have the myeloma cells, which are HGPRT negative and immunoglobulin negative in this step. So orally, you have the B cells from the mice, which will provide you the antibodies, and you also have the myeloma cells, which will help in division. When you fuse these both, you will get an hybridoma. Now, next step is cell fusion. Fusion of B cell with myeloma cell. Now, when you see after the third step, you have the activated B cells from the spleen of the mice, and you got the myeloma cells, which are HGPRT negative, and immunoglobulin negative. Now you're going to fuse these both mixture of cells by using a specialized chemical called PEPG, or simply you can say polyethylene glycol. Only for a few minutes, because prolonged fusion may lead to cytotoxicity, which we don't want. Now after fusion with polyethylene glycol or PEG, you have 5 types of cells. Those are fused B cells, fusion of 2 different B cells, which we don't want. Unfused B cells, which we don't want either. Fused myeloma cells, which are formed by the fusion of 2 myeloma cells. Unfused myeloma cells. And finally we have hybridoma, which are formed by the fusion of a B cell and a myeloma cell. Now we have to separate this hybridoma from this mixture of the B cells. Now you cannot separate justly by picking out. We have a method. So the next step is isolation of hybridoma. Now after the fusion, you have fused B cells, fusion of 2 different cells which we don't want. You have unfused B cells, which we don't want either. And you have fused myeloma cells, which we don't want. And finally also you have the unfused myeloma cells. But when you see, we have hybridoma cells, which we want because we have to isolate this hybridoma from the rest of the cell mixture. For this, you are going to put these all cells in one mixture called HAT medium. HAT stands for H for hypoxanthine, A for aminopterin, and T for thymidine. In this, the aminopterin will block the de novo pathway, which will not allow the cells to divide by de novo pathway. Now remember, B cells will have short lifespan, so they will naturally die. And here the myeloma cells are HGPRT negative, so they cannot synthesize by salvage pathway, so they also will die. The B cells will die and the myeloma cells will die. Only hybridoma cells will be alive because the B cell partner will supply HGPRT gene, which will help in the salvage pathway. So obviously they will survive. By the end of this, we have the bunch of hybridoma cells separated from the remaining cell mixture. They all will produce us the monoclonal antibodies. Now you all came to know that fusion of B cells with myeloma will form hybridoma. These all hybridomas are cultured in a specific separate medium. Now these all will produce antibodies only for a single epitope. These are called monoclonal antibodies, which will provide only for a single epitope. This is the overall procedure of hybridoma. Thank you for seeing this video. Share this Yakil. Thank you.